Yeah, I have 9:15 to 10:20. I have both my lectures.
I would just say, instead of I have mine
Hopefully your registration is going okay. There's a lot of registration anxiety. You have any questions about the bio aspect of it? I'd be happy to answer. Chemistry, nursing, I don't know how they run registration, so I probably won't have to answer.
But I probably won't be back.
Let's see.
I'm gonna have to finish.
How many of you registered today? A lot of you?
Yeah.
Yeah, I expected that. Based on the inbox of my email, I think a lot of people registered from these classes today. I think this was first year registration day. Let's see. Yeah, that's all I have about registration. Let me know if you have questions about bio aspects. Exams, I have not graded them yet because registration has been taking up all of my time. My goal is to get them to you next Tuesday, so you will have them before you leave for break. I'm going to try my absolute hardest to get that done. So fingers crossed there. Yes?
Is it going to be a case where we have with corrections to just bring it back the next week, or when we come back?
It'd be likely.
Okay.
Yeah. I haven't seen all the questions yet, so I don't know if there's corrections. I'm going to say likely, or how many. Yep. All right. Those are the announcements I had. But I did want to show you this slide, which I know you have as well, just about the remainder of the class, because we're kind of in the home stretch here, right? We have Thanksgiving break, and then we have, 2 or 3 weeks after that, and we're done. So what's on this slide is all that is remaining in terms of what you need to be aware of for deadlines in this class. All right. There is one 5-point assignment left. That's going to be a eugenics assignment. All right. It's very interesting. It's very enlightening. It's a very heavy assignment because we're talking about kind of a heavy topic. That has not been uploaded yet on Moodle. It will be shortly, so you can work ahead. The reason it's not up there yet is the video I used to have to watch is no longer available, so I have to find a new one. There are 4 content checks left. Right? One of them will be due before you leave for Thanksgiving break. The other 3 will be after. All right, so content check number 7 is due on Tuesday. Right? After lecture today, you'll be able to finish all but 1 question of it. And then as a reminder, your 2 lowest scores from those content checks are dropped. Right? So for both part 1 and part 2. Right? So if you got a 0 on 1 or 2 assignments, know that those zeros will be dropped. Scientific literacy assignment, right? That is up on Moodle. That is ready for you to begin. Get a head start on it, right? That is due after break. It is going to have you read a few papers which are posted on Moodle. One of them is a scientific paper. I don't expect you to fully understand the science that is in this paper. I just want you to get the gist of the paper, right? Enough that you could talk about it a little bit. Last day of class is the 16th of December, and then the final exam is the 18th, right? It's not in this room. It's in a different room. It's in Nobel 2220, which I believe is Wallenberg Auditorium. And it's an afternoon exam, right? So you'll be taking it from 3:30 to 5:30 in Wallenberg. So keep that in mind, put that on your calendar, whatever you need to do. The final exam is semi-cumulative, meaning that most of the material is going to be from this new unit. But there will be about 20 points about cumulative material. Right? After break, I will kind of inform you more on how I do that. But essentially, I'm going to give you some guidelines as to what to study for the cumulative material, so you're not totally blindsided. Right? Reminder with the exams, right? You have to pass at least one exam to pass the class, so keep that in mind as we're headed towards the final year. We talked about that on day 1, and it's been in the syllabus. And then if you have any conflicts with the final exam, right? You have one at the same time, or you have 3 in one day, having 3 in one day is grounds to move it around. And if you want to move to bio 1, let me know. Any questions? All right, so we're going to move on then and start the material. I know we're all still very excited and anxious about registration, but we're going to move on with the material, and we're going to talk about mutations, which is actually quite an interesting topic. We could spend a whole semester talking about mutations, but we only unfortunately have a few slides. So mutations. When we think of mutations, we often think they are a bad thing, right? They're not always necessarily bad. So mutation, we're talking about some change in the nucleotide sequence of our DNA, right? Some change has occurred, a change that is different than what it should be. They're not always bad. Sometimes they're beneficial. Right? Sometimes it's a mutation that actually increases the organism's ability to survive and reproduce. Right? So maybe it's a mutation that allows pollinators, say bees, to see the flowers more easily. Maybe it makes the flowers appear brighter to bees. Right? That flower is more likely going to be pollinated by those bees more often because it's easier to find, and that's beneficial for the plant. Right? That could be a beneficial mutation. That mutation might have no effect at all, right? So it's there, it occurred, but it's really not going to affect how the organism lives and reproduces. So for example, these tulips up at the top here, right? These tulips should all be red. There's been a mutation where half of one petal is now yellow, right? Likely that's not going to affect pollination, right? And likely this is not going to have any effect on its ability to survive and reproduce, right? So it's probably a neutral effect. Whereas sometimes we can get the bad mutations, the mutations that negatively affect an organism. These are the ones that are often talked about, right? And these are going to lower the organism's ability to survive. So the example I have on the board here is of a fruit fly, right? Here we see a normal fruit fly head. This fruit fly has had a mutation in its DNA that has caused, instead of an antenna to grow out of its head, it has a leg in place of the antenna, right? That's not very helpful. That's not useful. That's actually going to hinder this organism's ability to survive and reproduce. That is bad for the organism. All right, so big picture, mutations, not always a bad thing. Sometimes they're bad, sometimes they're good, sometimes they have no effect. All right, we're going to talk about how these mutations might occur and the types of mutations we might see occur. And we're going to talk first about these point mutations. So a point mutation is a change in the DNA code, right? Particularly at a single base pair. Right? So this might be where a base pair might be AT and now it's GC. Right? So that is a point mutation changing around the nucleotide in our genetic sequence. Now a point mutation might happen by itself. Might be just a singular point mutation in the genetic code. There could be multiple point mutations. Right? Maybe you have one point mutation and then directly next to it you have another point mutation. That could occur. It's more unlikely. There could be a mutation, a point mutation in one area of the genetic code. Then maybe hundreds of base pairs away, there's another point mutation. Right? So there could be multiple within our genetic sequence. And if a point mutation occurs, there's 3 different types that can happen. Right? It's either going to be a substitution mutation, an insertion mutation, or a deletion mutation. We're going to elaborate on this substitution mutation first. All right, so these are the 3 broad categories of mutations that can occur. And within each category, there are kind of like these subcategories defining those mutations a little bit more specifically. So a substitution mutation is where we are swapping out a nucleotide. Right? This is where maybe we have A, we're swapping it out to G. We're changing the nucleotide. Right? This can happen, it could happen spontaneously. Sometimes a cytosine will, it's called spontaneous deanimation, will change into a thymine. It could be that maybe there's a problem during DNA replication, right? Normally DNA replication is really accurate because DNA polymerase can proofread and correct itself, but maybe it missed something, right? Maybe that proofreading did not occur. And we get amino acids swapped out for something different. All right. So talking about our substitution mutations, we can have different types of substitution mutations. So we're going to talk about 4 different types, and I have images to go with almost all 4 of them. At the top here is going to be our normal segment of RNA, which translates to our normal strand of amino acids. Right? We call this the wild type allele, meaning it's the allele that is common. It's supposed to be that way. All right, we could have a mutation that changes out the nucleotide, right? So we're still changing the nucleotide, but it has no effect on the amino acid. All right, so an example of this, we have in this last stretch of our genetic code, we've gone from a codon that is GGC and codes for glycine to a codon that is GGU, but it still codes for glycine. Right? Because remember that genetic code, it's redundant. There are multiple codons that can code for a single amino acid. And in this case, the mutation occurred. It did change the nucleotide, but it still results in the same amino acid. Right? So it's not really going to have an effect on this protein. This protein will still be made as it should be. All right, so that would be a silent mutation. It's also called a synonymous mutation. Right? I like to use the word silent because it's easy to remember. It's a shorter word. We can have a missense mutation. Right? So this is where, again, we are changing a nucleotide in our genetic code. But this time that change in a nucleotide results in a codon that encodes for a completely different amino acid. Right? So here we've gone from GGC, which encodes for glycine, to now a mutation has occurred where we have AGC. And now it's no longer encoding for glycine, but it's encoding for serine. We have changed the amino acid that it encodes for. All right, so this can have an effect on the protein. Right? If we have a change like this could change how the protein is going to fold. Right? So remember, it's primary, secondary, tertiary, and quaternary structure. Right? This can have cascading effects on that 3-dimensional shape. And if shape determines function, that could change the function of this protein. We also have what's called a nonsense mutation. There's a lot of sense in these words. So nonsense mutation is going to change the amino acid from being an amino acid for encoding to an amino acid to now being a stop codon. All right, so here we're changing that second codon, which is AAG, typically encodes for lysine, to UAG, which is now a stop codon, which does not encode for an amino acid. So when this happens, right, we're going to stop translating prematurely. Right? We should have kept going, but instead we're stopping early. Right? So any codon beyond this stop codon is no longer going to be translated. All right? So this can shorten the protein. Right? We're basically deleting a chunk of amino acids out of the end. Again, could change the shape. could change the function. Our last type of substitution mutation is a sense mutation. Right, now I don't have a visual for this one, but this is where it would normally be a stop codon, and now it's going to mutate to encode for a regular amino acid. Right, so if we looked at this image at the top, we have UAA, that's our stop codon, right? If a sense mutation were to occur, right, this would now encode for an amino acid and would not stop. It would keep reading the mRNA until it reached another stop codon or until it ran out of mRNA to read. Right, so it's not stopping when it should. All right, lots of terms here and they all sound very similar. Science has to make it confusing for us, I know. I would know these terms and know if given a strand and a change has occurred, what type of mutation is it? And I'll give you some practice in a minute here. All right, so these are types of substitution mutations. Right, we are changing a nucleotide. This is where we already talked about this in the last unit. In fact, this was a question on the exam, right? But we talked about how there's redundancy in the genetic code, right? How there could be multiple codons encoding for the same amino acid, right? And this is why it's a good thing, right? Because if we had, say, UCU change into UCC, it would still be serine. It would be a silent mutation. It's not going to affect the functioning of the protein. Right, so often we call this third position the wobble position because it can change into any one of the three or four different nucleotides and still encode for the same amino acid. All right. Substitution mutations, so we're changing something. We also have insertion mutations and deletion mutations. Now we're not changing out a base pair, but rather we are adding in nucleotides or deleting out nucleotides. Right, so either we're adding in 1, 2, 3, so on nucleotides, we're adding it in, we're elongating our mRNA strand, or we're taking some out, we're shortening our mRNA strand. Now, when we have insertion or deletion mutations, this could lead to what's called a frame shift mutation. We'll talk about that in the next slide. All right, so we're either inserting extra nucleotides or we're deleting out some nucleotides that should have been kept. And that might result in a frame shift mutation. Right? This is when, if we add in nucleotides or delete out a number of nucleotides, it's going to change the way that we read our genetic code. All right, so we're going to do a non-science example first. Right? I have a sentence. Each word has 3 letters, just like each codon has 3 nucleotides. Right? So I have the fat cat sat. All right, makes sense. I can read that. I can encode that or decode that. If I were to take out that T, right, so I'm going to delete out that T, we're having a deletion here. It's going to change the way I read it because I still have to read it in groups of 3, but now it changes it completely. It's hef, at. It makes no sense, right? It's changing the way I read it. It's completely different now. We have shifted the way that we have read our codons or our letters in this case. All right, so let's go through a DNA sequence example. All right, we have our normal DNA sequence. It encodes for this amino acid sequence, GLN, PRO, THR. I'm going to insert a single nucleotide, right? So I'm not changing what's already there. I'm just inserting an extra one. That's changing the way that I group my codons. right? So the first one is still going to read normally, still going to be GLN, but now I'm going to change how I read the rest of it. So now I have serine and histine, which I did not have before, because that's what my codons now tell me it encodes for. It's changing the way I'm reading it. Right? It's shifting the way that I'm reading this DNA sequence. Notice it also shifts it so there's only one nucleotide in the fourth codon instead of having three. Right? That's kind of nonsensical. It's not going to encode for really anything. All right, so that's an example if I added in 1. What happens if I added in 3? Because codons aren't sets of 3. Right? Well, it's still going to lead to a change, but it's not going to be a frame shift. Right? So I'm adding in 3 in a row. My first one is still going to be the same. I'm adding in this extra amino acid. And then after those 3, it's still going to be the same. I'm not shifting how I read my genetic code. I just have an extra amino acid encoded for it because it was a multiple of 3, and we read codons in multiples of 3. All right, let's do another example. So I have the normal wild type allele at the top here, what I should see. Right, if I either insert or delete out nucleotides, and that insertion or deletion is not in a multiple of 3, I'm going to get a frame shift mutation. It's going to change how I read the genetic code. But if that insertion or deletion is in a multiple of 3, I'm either going to have one extra amino acid, right, or delete out an amino acid. It's not a frame shift mutation, right? It's just changing the amino acids. All right, one more slide and then we're going to practice. I've developed this flow chart to help you out here. So we're going to say a mutation has occurred. The first question you are going to ask to figure out what type of mutation it is are the nucleotides changed or are the nucleotides added or removed? If they're changed, we know it's a substitution mutation. We would then ask the question, did that change result in the same amino acid, a different amino acid, a stop codon, or removing a stop codon? And that will tell me what type of substitution mutation. All right, now if we went back up to the top and said nucleotides are not changed, but they're added or removed, we would know that this would be an insertion or a deletion event. And then we'd have to look to see if the nucleotides were removed in groups of 3 or added or not removed, or added or removed not in groups of 3. Right? It's that groups of 3 that is the key here. And that will tell us whether or not a frame shift has also occurred during this insertion or deletion. All right, let's practice. So I have given you the original RNA strand. I want you to tell me for each of these mutations that I've highlighted, what is the name of this type of mutation, and what is the consequence of this mutation? Oh, don't want to give you the answers yet.
So it switches over. Yeah, it's switching from face and not. So it's just changing. Oh, yes, and now it's quiet. It's silent. It doesn't change the name. Okay, the next one. It's also substitution.
Yeah, it's not gonna solve it, so it's the
So silent.
no. It's the same. It's not the same. So it would be missing.
Yes, it's actually.
The next one.
Still a substitute. So still missing. Oh, it's a stop. It's a stop. Oh, yeah, it's a nonsense mutation. The last one.
Is it insertion?
Yeah.
With the frame shift. increase to at the bottom?
It does. Let's see that.
So if you're on a wait list, it's either if they increase the cap and you're next in line, essentially, or if a student moves their schedule and then a slot. So
Okay.
it's hard to predict when.
Okay.
Yeah.
Thank you.
So in here, good in terms of bio class, maybe not in terms of registration.
Yeah, I don't know.
We got rapper and actor.
I wonder which kind of mutation causes my mutation.
Like that could be a little investigative report, and then maybe get some extra credit.
No. They know for all of the other reasons my disorder, because there's like subsets, just not the exact type I have. It's the only one.
You're special.
I am special. I also just happen to be the group that has the most, just most people who have
Yeah.
1% of the population. It's a lot. It is.
Have you done like DNA analyses of your genes?
No, but my mom.
Yeah. That'd be expensive. It's getting cheaper and cheaper by the year.
Doing a gene analysis wouldn't help. I don't know what gene causes it either.
No, yeah. So you have to do like the whole genome and that's lengthy and expensive.
Great idea. Fun plans for Thanksgiving?
Go home, see my family? Okay. Yeah.
Some family excited here? You're excited to see?
Yeah.
Okay.
Yeah. Get to go home to see my side of the family this time, which is nice.
Yes. You get to suffer next year?
I might. We'll see. The other side of my family is intense. Yes. We actually had early Thanksgiving this past weekend, so.
Nice.
Yikes.
Are they about the same size?
Okay. Yeah.
Do you cook for Thanksgiving?
I'll bring like a charcuterie board.
Okay.
I've had to cook for Thanksgiving since I was like 8.
Yeah.
You're looking at these. Always
My dad's the cook. He always makes an amazing meal. So.
I wrote in my feelings at least one thing.
Yeah.
Changed. My dad probably cooks all the shit.
It doesn't matter how many it is.
That's what we should do. Everyone cooks one thing.
I
Yeah, bring
cook
us
2
an eye.
things and then I
Like,
do
I'm gonna go back.
like
It's either,
the tray.
is it Ingrid?
Okay.
But I do the mashed potatoes and those.
My dad is very like stuck to tradition. This is what we've had in the past. We're keep having the same meal, same recipes.
If it makes it
I don't use a recipe.
Well, no, the first one.
Yeah.
Just different every time I'm making
Every year gets better and better.
it. It does. I'm so
Right, but
Dude, we did the food
they're both the
from
same class.
like 20
So it's
pounds.
if you choose to,
It was
but then the
absurd.
second one.
Last year someone burned our mashed potatoes, so this year they're bringing soda. They've been demoted.
Last one I was adding.
My dad does the turkey and the stuffing and pies. Different. My mom does various things. She does a lot.
All right, let's go through these examples. I learned from my last class that as soon as I press my clicker, all the answers are going to show. This first one, all right, what type of mutation is it? Is it a substitution or insertion or deletion? It's a substitution. What type of substitution? It's a silent, yes, because what is the consequence? What is happening to the amino acids? Yeah, nothing, right? Even though we've changed our nucleotide, we haven't changed the resulting amino acid. All right, what about the second one? What type of mutation? Substitution and missense. Yeah, because now we're changing the amino acid. All right, I'm going to press my button now and you'll get all the answers. Except the first one did not work in my animation. That should say silent, but it's not unveiling for you. That second one is a missense, right? That change has resulted in a change of amino acid. We've gone from the TRP amino acid to the TYR amino acid. All right, hopefully this next one you saw is a nonsense. Right, we've gone from an amino acid to now it being a stop codon. So we're stopping prematurely. And then this last one, I actually didn't change the original strand. I just added in a couple of extras. Right, and I added it in a group of 2, which means not only is it an insertion, it's also going to cause a frame shift. All right, so it's going to change all of those amino acids after the insertion event. Any questions about these mutations? All right, like I said, mutations are fascinating. We could spend a whole semester on them. I only have 3 more slides. So, and if you want, there you go. So far we've been talking about a mutation that happens within the coding region of a gene. Right? A mutation that happens in the area that gets translated into an amino acid. Right? But we know from the last unit that not all areas of our genetic code are the coding area. Not all areas directly encode for an amino acid. We have that promoter region, we have the terminator region, we have some introns. We can get mutations in those areas as well. Right? So if we have a mutation in the promoter region, it might increase or decrease the rate of transcription. Right? It might encode for faster transcription or slower transcription. Now there's typically some elements, some proteins or other molecules that help regulate this transcription beginning. Right? We can get mutations in those elements, so that might alter how transcription works, like how much is expressed, when it's expressed. Maybe it's saying to transcribe this gene, even though we don't need that gene being made for that particular cell. Splice sites, maybe the changes in the mRNA. We've had a mutation around our splicing sites, trying to get rid of our introns. And maybe now we're not getting rid of all of our introns. Or we're getting rid of too much, right? The introns plus some of the extrons. Right? It's going to cause some problems, so we're not properly splicing our gene. And then translation. When translation occurs, right, there's other proteins and elements in play. We can also get a mutation in those. So that might alter the ability to take RNA and translate it into an amino acid sequence. Right, so big picture on this slide is that mutations can happen anywhere in our DNA. It doesn't have to be in the region that encodes for an amino acid. It can be outside of that region, right? Promoter, terminator, within elements that affect how fast it goes, if we can translate, all sorts of mutations. There's a lot of text on this slide. All right, so many different types of mutations can occur. Mutations can also occur in different types of cells. Right, so they might happen in the germline cells. So these are cells that will go through meiosis and give rise to gametes. Right? So if this happens, the mutation is going to show up in the gamete. Right? And if that mutated gene gets fertilized, right, that mutated gamete gets fertilized by another gamete, it has the possibility of passing that mutation on to its offspring. Right? And if that mutation gets passed on to its offspring, that means the entire individual that results from that offspring is going to carry that mutation. It's going to affect the entire individual. Right? But we can also have mutations in somatic cells. Right? So these are all the other cells beyond gametes. And when that happens, typically that mutation is only going to affect a portion of the individual, right? Or the area where that mutation started. Right? So say we had a mutation, right? We have this developing embryo That mutation is going to be locally centralized when that embryo develops into a full organism, right? Again, it's still locally centralized. We're only going to see mutations in that patch of area. And because somatic cells are not part of the germline, right? They're not going to develop or create gametes. This mutation is not going to be passed down to offspring, right? So it's not going to affect the gametes. It's not going to have the ability to pass it down. All right, so it can happen in gametes. It can happen in pretty much any cell, right? And therefore, it's going to affect any offspring differently if it affects it at all. So what actually causes these mutations, right? Why do these mutations even exist? Well, mutations can be spontaneous, right? Or they can be the cause of some biological process going awry, right? So maybe it's errors in DNA replication. Maybe that DNA polymerase was not able to proofread itself, right? And we had an error. Or maybe there's an insertion or deletion event after DNA replication, right? Something's gone wrong biologically. It also might be that there might be other byproducts in our bodies that can affect our DNA. So when we talked about fermentation, we talked about the problem with fermentation is getting too much NADH, right? And too much NADH, right? Those NADH molecules are going to start randomly giving electrons away. That's going to create what's called free radicals. And those free radicals can damage our DNA, leading to mutations. Right? So toxic byproducts can also be a biological cause. There can also be causes that are not biological, that are due to the environment that we are in. Right? Typically, this is like a physical or a chemical cause. Right? So I have two examples up here. My first example is a chemical cause. Right? So benzoapyrene is found in cigarette smoke. Right? And that chemical actually jams itself in the bases between the bases of our DNA. Right? And that's going to cause insertions or deletions. Right? So that can cause a mutation. Right? There are other chemical mutations that are possible. You might have heard about a few of them in the news or in other examples. You could do a Google search and find many examples of environmental disasters, particularly, might be causing genetic mutations within a population. Maybe you've heard of an environmental disaster and all the babies are born with some rare disease or have a higher chance of cancers, right? That is environmentally induced. But there can also be physically induced changes, right? Like X-rays. If you get an X-ray, right? X-rays have the potential to cause base deletions by breaking the DNA, right? So that's why if you get an X-ray, they typically put this protective covering over the area that is getting an X-ray taken of it. So if it's your leg is covered with an extra mat, and that's to help protect against these X-rays causing damage to your DNA. That's why the technicians, the radiologists often go into another room or behind a screen because they're around X-rays so often, they don't want to expose themselves to potential effects of mutations by X-rays. Right? Sunlight is also an example of this, right? UV radiation from the sun. You've heard of make sure you wear your sunscreen, because if you don't, sometimes you can get skin cancers because of that UV radiation. It can cause mutations. Right? So there's a lot of causes of mutations. All right, any questions about mutations before we move on? I'm going to take that as a no. All right, so we've talked about DNA, we've talked about mutations and how mutations can get passed on, but we haven't talked about inheritance patterns, right? So this is where we're going to start talking about Punnett squares and Gregor Mendel and his pea plants. All right, so we're going to look at Mendelian inheritance, called so because of Gregor Mendel. my first slide here should contain information that you already know, but I wanted to explicitly state the definitions of trait and heredity. All right, so if we're talking about passing along genetic information, we're talking about storing information in our DNA, and our DNA is packaged into chromosomes, and those chromosomes can be passed along to our offspring. So a trait is simply any characteristic of an individual, right? Eye color, hair color, right? Maybe it's a certain way that your body processes food, right? Maybe it's a trait that you inherited some like intolerance to a food. Whereas heredity is how that trait is passed on, right? The transmission or the passing on of that trait from the parents to the offspring. Right, again, this shouldn't be new information. We've kind of been talking about this, but I did want to explicitly put these definitions up here. All right, so if we're talking about Mendelian inheritance, we have to talk about Gregor Mendel. Right? Gregor Mendel was a scientist in the 1800s, right? And he was really interested into how do traits get passed on? Why is it that we can look at an organism or say a human and they look kind of like their parents, right? What is that process of passing along the traits? How does this happen? So he did his experiments on plants, particularly pea plants, which we'll talk about next. And he published the results of his studies. He published it in the Proceedings of the Natural History Society of Brunn. Right? So his science was out there. Now it's interesting if we're thinking like historical context. Gregor Mendel and Charles Darwin were actually alive at the same time, and they were exploring the same type of questions. Right? Gregor Mendel was talking about how do we inherit traits. Darwin was talking about how do species evolve, so how do traits get inherited and change over time. Right? But they didn't know about each other. Right? They were unaware of each other's work. So they did not collaborate together, but this research was happening at the same time. Now, unfortunately for Mendel, right, he published his studies, but his studies were actually kind of lost. No one really cared about it. It was lost. It was buried. It wasn't until in 1900 that we rediscovered his discoveries, right, and we're able to see how much Gregor Mendel had actually discovered about inheritance. All right, so Gregor Mendel famously worked with pea plants. And in this case, Gregor Mendel got really lucky. All right, so Gregor Mendel, again, he was asking these questions. Why do the offspring resemble their parents? And how does this passing along of traits occur? All right, so why does this cute little puppy look like its parent? Right? Why is that? How does it occur? So he looked at his pea plants, right? And again, he got lucky because a pea plant is a model organism, right? You might hear that throughout your biological career, right? Something is a model organism. And a model organism basically means it's really great to work with. It's easy to work with for a variety of ways, right? So pea plants are a model organism. Nowadays, if we're looking at genetics, typically scientists will use a plant called Arabidopsis because that's a model organism for how fast it grows and how easy it is to take care of. We actually have a professor in the department that works with Arabidopsis plants. Fruit flies, they're another example of a model organism. Again, they're easy to care for, right? And they have traits that we can easily observe. But pea plants were a model organism for a variety of means. It's easy to grow, right? They're not hard to grow. All you have to do is find a pea pod, right, or a pea seed, stick it in the soil, add some water, give it some sunlight, it's going to grow. It's not a finicky plant. Right? Its reproductive cycle is also very short. Right? It can flower within a matter of weeks. Right? So we're going to get new individuals, or at least the precursors, seeds, to new individuals within a matter of weeks or months. Right? So it's fast. Take, for example, if you were studying elephants. right? If you were to look at a baby elephant, right, and then look at the baby of that baby elephant, we're talking on a timeline of years, right? Years to see different generations. This is only weeks or months. It also produces a large number of offspring, right? So it produces many seeds, not just one or two. Again, elephants would be one, maybe two, but tens to hundreds of seeds per plant. Lots of offspring. It has traits that are easy to see, right? So if you look at a pea plant, you can see the color of the flower, right? You could see the color of the seeds or the shape of the seeds. Traits are easy to see. And finally, the mating of these plants are really easy to control, right? We can control which plant is mated with another plant. All right, so what I mean by that is we can do artificial pollination here. And the way that this is typically done in science is with a paintbrush. You would isolate the plants so that no pollinators can get in. We're making sure bees cannot pollinate our plants. Only we can pollinate our plants. And we're going to pollinate our plants using a paintbrush. We're going to use the paintbrush to brush pollen onto our paintbrush, and then bring it over to the plant that we want to be the other parent, and take that pollen off, right? Brush it onto the stigma or the carpel of the flower, the female flower parts. And then we can control. We know which one was the male. We know which one was the female, so the mother and the father. We know who the mating pair was, right? So kind of a side note to this, paintbrushes are often used. I've also seen it in science where they take a bee, it's a dead bee, they put it on like a needle, and they brush this dead bee against each flower to try to mimic how a bee is going to move and vibrate. It's very gruesome looking, right? But I have seen it done. Paintbrushes are far more common. If I have a plant in my garden that is struggling to reproduce, I will often take a paintbrush and try to make sure they get the pollen. So what results, right, is a plant or a series of plants, all of the offspring, who we know were the mom and dad, right? We know their genetic line. Now, often when we see examples of inheritance, we might often see the offspring labeled as like F1 or F2. And you're going to see this quite a bit, so I wanted to call it out, right? F1 literally means first allele, which is translated to mean son or daughter. So the first sons and daughters of this generation, right? We would call this the F1 generation. All right, so pea plants. They're great because they're a model organism. Mendel got lucky. He again got lucky in that the traits that he was looking at only had 2 different versions for each trait. All right, so he looked at a variety of traits. So for seed shape, it was either round or wrinkled. There wasn't a third option. There wasn't a fourth option. Just 2 options. And there were only 2 options for all of the traits that he studied. So he studied everything from color to shape, right, to how the flowers grew on the plants, to how the plants grew. Did they grow tall or did they grow short? Right, so he observed these features, these traits, and these observable features we would call a phenotype. Right, so he looked at their phenotypes. He looked to see if their phenotype was round or wrinkled for seed shape. He looked to see if their phenotype was purple or white for flower color. Okay. And again, he got lucky in that there are only 2 distinct phenotypes for each trait. Right, so he picked a great plant. This plant was super easy to study. All right. So we're looking at phenotype, the physical characteristics. That's how I like to remember it. Phenotype, physical characteristics. We can physically see it, right? Physical starts with a P, phenotype starts with a P. All right, so he worked with these pea plants and he started crossing them. And he started working with what we call pure lines, meaning each plant, we call them a true breeding pair, right? So like the male and the female parents, they only had the genetics for one trait, right? So the male parents, right, only had the genetics to pass along the alleles for round seeds, right? And the female parents only had the genetics to pass down the alleles for wrinkled seeds, right? Because they only have those genetics, we call them pure lines, right? They don't have two different versions of an allele, they only have one version. He would cross these pure lines together to create a hybrid, right? Or the first generation, the F1 generation. And Mendel noticed something when he crossed these plants. Something odd happened, right? That first generation only showed one phenotype, even though he crossed parents that had two different phenotypes. Right? That first generation only showed one phenotype. All right, so that's a little odd. So he kept experimenting, right? So he crossed his true breeding lines. He got this first generation where they all looked alike. Only one phenotype was present. He then crossed two plants from this F1 generation to get the F2 generation, right? So the second generation. And in the second generation, he also noticed something odd, right? The wrinkled pea, that phenotype, now showed back up. Even though he crossed two plants that did not have the phenotype of showing wrinkled pea seeds. Right? So now this phenotype is reappearing. Not only is it reappearing, but it's reappearing in this very specific ratio of 3:1. So 3/4 of the offspring would have this smooth pea pod, and 1/4 would have this wrinkled pea pod. Right? So this 3:1 ratio was appearing in this second generation, this F2 generation. All right, so that's a pattern we're starting to see. It's very unique. It's kind of odd. You might question it, which is what Gregor Mendel did. Right? Not only did that happen in a seed shape, but it happened for all of his traits. In the F2 generation, for all of his traits, you saw this 3:1 ratio. Right? 3/4 were a certain color or a certain shape, and 1/4 was the opposite phenotype. All right, so this pattern is emerging. So based on this pattern, Mendel was able to determine that some traits are dominant and some traits are recessive. Right? So a dominant trait is a trait that, if it's present, will always be expressed. Right? If you have one allele out of two that is dominant, the dominant allele is going to be expressed. If both alleles are dominant, the dominant allele is going to be expressed. If it's present, you're going to see it. Right? So that F1 generation produced round seeds. So we would assume for this F1 generation, it had the alleles, all the individuals had at least one allele that was dominant, because that's the trait we're seeing. Now, a recessive allele is a trait that can be masked by the dominant. Right? So if we have an allele that's dominant and recessive, right? So we have a homologous pair, one has the dominant trait, one has the recessive trait. The dominant trait is always going to mask the expression of the recessive. Right? That recessive phenotype is not going to show. But if we have two alleles that are recessive, we don't have a dominant allele at all, then the recessive phenotype can show. Right? And that's what he saw in that second generation. He saw that some of the pea pods showed the dominant phenotype, and thus must have a dominant allele. And you saw a few of them had this recessive phenotype, and thus must not have any dominant alleles. Right? So if there is a dominant allele present, it will always be expressed. All right, I have talked a lot, so this is a quick break for us all. I want you to answer these 3 questions, and then we'll move on to some Punnett squares.
Because, oh my god. They're just a fish.
It's a trait that's shown. Okay. The one that pops up. Okay. Yeah. The one you can see. The eyeball. Why would that?
Whatever, 6th grade.
Science was great. Actually, my favorite.
Do you watch Gaten?
No. I can start.
Okay.
I was like,
Reading is what got me into honor science in middle school. I was dominating this. They were like, it's tough for you. And I felt so fancy.
No. You were always
No, we're just distracted by registration.
I was 12,
But don't worry. No, but I do want to physical trait.
questions here at all.
Flies. It made me think of a friend.
Now we got it.
I remember I was in high school, I guess that's
Dementia-induced death.
Oh, this is what it looks like.
Tried it so much dementia made it
Eat by eat.
And the results said all the people that died.
Oh, we will see. Yeah, that's what you backed across.
Did you give them this
Oh,
lecture?
okay. I'm guessing that's Tuesday,
That's Tuesday. Yep.
I could have predicted.
Yeah, well, I don't know. I guess you probably you're right.
Also, fruit flies die fast as fuck.
Right.
It's like a bad.
Yeah, it was quick.
At least it's an ethical choice. Something that's
Right, because she wanted to use wraps, but they wouldn't.
Oh,
yeah, because that's evil. They have the evil.
Okay, good.
I have nothing to change. You fought for him. They were like, no, we're not like
Your friend is evil.
She's got a little spark of
not you.
Satan.
You know what She tried to commit animal abuse.
Say is to your parents who are not biologists, what would you tell them is a phenotype? Physical characteristics, it's what you see. Yes, it's what you see on organisms. Right? So my phenotype would be this blonde hair, right? Or that I'm kind of short. Right? That would be what I see. That is my phenotype. All right, why were pea plants an ideal organism? Yeah, there's only 2 observable traits for each. Yeah, quick and easy. They're cheap. It's not hard to get a hold of pea seeds. Yeah, I mean, there's a bunch of characteristics that made it great. All right, so now if I were to breed together 2 true breeding organisms for 1 trait, how would I tell the dominant trait if I were to look at their offspring? Yeah, I'd do their phenotype. Yeah, so if I have 2 true breeding lines, I breed them, I look at their offspring, they should all have the same trait in that offspring line, in that F1 generation. And that trait that we are seeing would be the dominant trait. All right, we'll keep emphasizing this. All right. More terminology, but it's terminology you already know. I didn't even take away the blanks in your slides because you should know these definitions. But what I do want to emphasize here is that Mendel didn't know about genes, right? The word gene was not a thing, right? He knew what it was, but there wasn't this word to put to it, right? So we now know that our genetic code is what determines our gene, right? Mendel, though, saw these genes and saw that there were different traits associated with it and came up with the idea that there were different versions, or what we now know as alleles, right? And it was these different versions that were responsible for the phenotype. Right, so again, I put another example. I'm trying to use different types of organisms for my examples here. But fruit flies, they have a gene for eye color, right? They can have different eye colors. One is red, one is purple, right? The different versions are the alleles. All right, we should be familiar with those terms. But this term is one that you might not be familiar with, or at least we haven't covered in our class yet. So the alleles found within an individual are called its genotype. Right? Together, those alleles we would say is its genotype. Right? So if we put version 1, or if it has two versions, right? Version 1 and version 2, that's its genotype. Maybe it only has one version, but two copies of it. Version 1, version 1. That's its genotype. And the genotype is what determines the phenotype, or what it appears to look like. All right, so let's walk through an example, just to kind of solidify this. Here we have a pair of homologous chromosomes. We're specifically looking at the gene for height. It has two different versions of this gene, right? One version is the tall allele, one version is the dwarf allele. Now the tall allele, right, is capital T, right? And it's a capital letter because it's the dominant allele. The dwarf allele is going to be the lowercase letter of the dominant trait. So it's going to be lowercase t. And together, capital T, lowercase t, that is the genotype. All right? So if we're looking at our genotype, capital T, lowercase t, we know we have at least one instance of a dominant allele. And because we know if there's at least one instance of a dominant allele, that's the allele that's going to show up in the phenotype. Right? So here we have possible phenotypes. So for this particular genotype, which phenotype should we see being expressed? Yeah, it should be tall, because we have at least one allele, that is the dominant allele. Right? For this genotype, we would expect the tall phenotype. All right, we're going to keep practicing this. In fact, we're going to have practice now. So I want you to use this information and use the letter P, right? The letter P would be our different alleles, either capital P or lowercase p, right? And tell me, what would the phenotype be? And then what would the genotype be? So remember, the phenotype is what you see. We're not going to use P's here.
So the phenotype is that it's purple color, and the genotype is big P, little p, or big P, big P. And then the recessive one is okay. So genotypes are little p, little p.
What is your favorite flower?
Lily of the Valley.
Not the way it smells.
I don't smell flowers much.
No.
I just look how it looks.
It's a very pretty flower.
And then how's it going?
Good.
Yeah, it's a registration stress.
I didn't get my chem class. I don't know how to fix that. Or
Is it more organic chemistry?
yeah. Like baby steps.
Yeah, for.
And then like I bought you flowers.
That's really funny.
And I remember somebody in the department.
That's what genuinely so like
My friend, don't flowers.
And so I like any of them or the one that works for my schedule.
I would genuinely be like, that's so funny, thank you.
Because I don't know how chemistry teaches it. I would start by either emailing someone whose section you want, or maybe your current professor. Yeah, they'll get you to the right person.
I don't want to plan this. I have to put in work for these. I wouldn't be mad if I just be like, damn, I'm not. These things are going in the closet. They're gonna grow mold. Flower seeds.
Brandon, would you rather get flower seeds, a live flower bouquet, or different types of bags of flour?
Like the cookies.
Yeah,
The cooking item.
yeah.
I want cut flowers.
Okay.
They're pretty and
I
they're
said bags
immediate.
of flour.
But seeds might be a bad gift.
Yeah.
I personally just don't want the effort.
Yeah. The flowers are fun because you can grow flowers that maybe you, or you have flowers that maybe are harder to grow
Yeah.
or out of season.
I'd press it.
It has an environmental impact, but
But we're
I'd
celebrating.
press the cut butter to dry.
all right. So depending on your background in biology, this question may have been easy. But let's start with the phenotype. The phenotype of this top flower, what is it? Purple color, yes, purple flowers. All right, phenotype of the bottom flower, white flowers, great. What would be the genotype for my dominant flower, my purple flowers?
Big P, little P, big P, big P.
Yeah, so it could be both uppercase P's, but it could have been one of each, right? We don't know for sure what the genotype is. We just know there has to be at least one dominant allele in order for that dominant trait to be expressed. All right, so white flowers, what is the genotype here? Little b, yes. It can only have the recessive alleles in order for the recessive phenotype to show up. All right, so this leads us into, yes, more terminology, but terminology you're probably familiar with. Right, so when we're talking about genotype, we could say that genotypes are homozygous or heterozygous. And that depends on what type of alleles or mixture of alleles are present. Right? If it's a homozygous individual for that trait, it means it only has one version of the allele. Right? It only has 2 copies of the dominant allele or 2 copies of the recessive allele. Right, so in this case, both our dominant and our recessive flowers have the possibility to be homozygous individuals for flower color. Either it could be homozygous dominant, so have 2 dominant alleles, or homozygous recessive, and have 2 recessive alleles. Right, now the purple flowers could also have one of each, and we could say it's heterozygous. Right, there's a mixture of alleles. Right. It carries 2 different versions for a given gene. So it has the dominant allele, but it also has the recessive allele. What's going to be expressed in the phenotype, though, is the dominant because it masks the recessive. All right. Let's talk about Punnett squares. So we're going to keep thinking about that idea about breeding true breeding pairs. Right, getting an F1 generation, and then breeding that F1 generation to get an F2 generation. And so we're going to look at a cross between 2 homozygotes for the trait pea seed color. Right, so these would be true breeding individuals because they only carry one trait. Right, still have 2 alleles, but those 2 alleles are the same. Right, so one individual is going to be homozygous dominant, and that is our yellow seed, so capital Y, capital Y. Right, so when it goes through meiosis, it has the possibility, I'm going to let it go. All right, there we go. It has the possibility here of creating gametes, right, but the gametes are only going to be able to carry down a capital Y or a capital Y. So for each column, we're going to put a capital Y. The other true breeding individual is homozygous recessive, so it has 2 recessive alleles, only has the possibility of passing down a recessive trait, right? So it can only be a lowercase y being passed down or a lowercase y being passed down. We would label each row. Then it's a matter of matching these rows and columns together. Right, so this first row and column, we would get capital Y, lowercase y. And in fact, we would get that for every time we tried to make a match here. All right, so we've taken two true breeding individuals. We've created the F1 generation. And the F1 generation all looks the same. And we should expect that, right? We know that's what happens with an F1 generation. And this is the mechanisms, right? The genotypes working to make sure that the F1 generation all look the same. All right, they're all yellow seeds. They all have at least one dominant allele. All right, so now we're going to take two of these individuals from our F1 generation and mate them together. Right? So we're going to do a cross between two heterozygotes. Right? We're crossing two individuals from the F1 generation. They're two heterozygotes, so they're going to have a capital and a lowercase letter. They're going to have a dominant and a recessive allele. Again, we're going to match them up, right? And we're going to see something occurring where we get that ratio we talked about, that 3-to-1 ratio. Right? Now, if we're looking at color, we're talking about the phenotypic ratio. And that 3-to-1 ratio is what we are seeing, right? 3 of them are going to show up as yellow because they have at least one dominant allele. One of the possibilities is going to be green, right, because they have both of the recessive. Right, you could also say 3/4 are yellow and 1/4 are green. Right, get that 3-to-1 ratio. All right, so this is what Mendel observed for all of his pea plants. Now that we know the genotypes of these individuals, or the possible genotypes, we could also look at the genotypic ratio. All right, so now instead of focusing on the phenotype, the color, we're looking at the genotype, or the letters. We see there's one instance of being heterozygous dominant, capital Y, capital Y. There's 2 instances of being heterozygous, so one of each. And there's 1 instance of being homozygous recessive. Right, so we could say it's a 1-to-2-to-1 ratio. Right, 1/4 are homozygous dominant, 1/4 are homozygous recessive, and 2/4, or 1/2, if we reduce the fraction, is heterozygous. That would be our genotypic ratio. So again, only looking at genotype here. Phenotypic ratio, only looking at what we see. All right. I believe my next slide is practice. So let's do a review. I want you to tell me what are the genotypes for my true breeding parents. What is the genotype for the F1 and the F2 generation? And then tell me the phenotypic and genotypic ratios for the F2 generation. You can use a Punnett square if that helps you.
Big P, big P, little p, little p. Second, your first generation,
Yeah.
is big P, little p. It's heterozygous. And then the second generation, big P, 2 big P, little p's, and look at P. So it's 1, 2, 1. And then the phenotypic ratio is 3.
What they call it, do you remember?
Yeah. How often is this being taught, and then they do eye colors, and then kids find out that they're not their parents' kids?
You would be surprised, it's a lot.
Like,
But also, sometimes they'll do it with hair,
You let me
and they don't realize that there's more to hair than just like dominant.
okay.
And so people are like, oh, it's genetic.
Genotype, yeah.
Yeah. Yeah, so they'll be like, okay, there's no way that can happen. Your parents cheated, and then they didn't. And it's like, you can't say that to a student. You're a liar. Also, you should just don't say that. Like,
Let's just focus on flowers.
that's why people do the earlobe hitchhiker stuff. If you have 6 fingers,
Oh.
because that's the
So
bone.
the genotypes of the true breeding generation. It would be too bad.
The one polydactyl was the other one, that got their extra.
That generation would be.
Yeah, it's so weird.
Because the
You
other
could've won in every family.
flower, the other true breeding.
I don't know if
but
that's how that works.
genotype.
Unless
I
they
know
have
the
2
thumb
thumbs.
Don't they
Use
usually
the
just
white
fill
one
it?
there.
Or you just pinky? Oh. Oh, I've seen extra fellow.
If I had an extra thumb, I'd go wrong.
You're part of the polydactyl with the eggs.
It's like a little hook.
Same like that.
Most people have polydactyly. I don't think they know.
I have never met someone with more than 10 and less than 10. Or really
I haven't either, but I could really change this.
F1 generation has always been like when we're talking about a true
What
breeding.
about the people who have their the skip and
Oh, the web?
Yeah. Or like their feet are hurt, so they have to get
So
like
once you figured out, so I'm guessing you're what you're telling her, like, this is double, like, capital, and this one's double.
like
I'm pretty close to that, but
if I had webbed fingers, I would be
So when you make your mix, you're trying to figure out what of the 4 options would be aligned.
Michael Phelps would go crazy.
Big, a little bit.
He already has genetic.
Does that make sense?
He doesn't
Most
And then
Olympians.
that's,
Yeah. But he has a fast food
if
time because
I mean, they're saying it's, the breeding generation.
Oh.
Also, he has absurdly
Right, because then even if this is.
how tall is my development?
Alright, let's go through these answers
If
here.
I was in the Olympics, though,
If I'm
you
talking
could
about true breeding individuals for the
so no
parents,
drag.
what is the genotype for my pink
I'd be
flower?
I have no eyebrows.
2 uppercase P's, yeah. So capital P, capital P. Alright, what about for my other parents? Well, I gave you the answer. It would be lowercase p, lowercase p. Right? For breeding, they only have one version of the allele. But they still have homologous pairs, so there's still 2 copies. Alright, so they're going to have cross-fertilization happening. And they're going to come up with the F1 generation, which has the genotype of what? Big P, little p, yes. Because it needs 1 allele from each mom and dad, and the only possibility is to be a heterozygote. Alright. And then they're going to self-fertilize, right, or be pollinated within the generation. And we're going to get more offspring in the F2 generation. What could be the possible genotypes for my pink flowers? Yes, we're going to get either homozygous dominant or recessive, right, or a heterozygote. How many of these are going to be heterozygotes? 2. Yeah, so 2 of them. If you did that Punnett square, you'll see it's the 1 to 2 to 1 genotypic ratio here, right, with this Punnett square. All right, I kind of gave you the answer for the genotypic ratio. What is the phenotypic ratio? 3 to 1. Yeah, that's what we're physically seeing. 3 are pink, 1 is white. Right? So it'd be a 3 to 1 ratio. Then again, if you did that Punnett square, you found the possibility of the genotypes for the F2 generation. You would see it's a 1 to 2 to 1 ratio. All right. You're going to get out early because this is where my last class ended. Homework, there is the content check due on Tuesday. You'll be able to do all but 1 question. You can work ahead on some others. Is it not open yet? Did I have it set to like 1 o'clock or something?
I tried to do it.
Yeah.
I worked on it.